As I am Dr. Anshu Singh, I have done my MTech PhD from IIT Kharagpur, and I will be your instructor for this webinar. Okay. So the sessions has been divided into the 4 sections. First, we will be talking about the concept of industrial biotechnology. Then what is the potential of industrial biotechnology? Certain case studies which are related to the application of industrial biotechnology. And then we will be talking about scope and career opportunities related to this industrial biotechnology. So you all know that at global scale, we are facing the climate crisis, okay? And the way we are heading forward, we are spinning out of control And at the same time, you can see our world is facing the major pandemic crisis, okay? By 2050, our global human population will reach to 9 million, okay? And majority of people will be below poverty line if we fail to alter our current development pathways, okay? So what is an option left where we can meet our demands, we can hold our resources, natural resources, and we can improve our climatic conditions, okay? The option is greener approach, okay? And the greener approach options opens an avenue for industrial biotechnology, okay? So you all know when we talk about industrial, it is related to industry, that is scaling a process. And when we talk about biotechnology, you know, the biotechnology is development of a bio-based product using living cells such as yeast, fungi, bacteria, plants, and enzymes. And when we use them at industrial scale, that term is called industrial biotechnology, okay? The question arises, to what extent industrial biotechnology can transform a fundamentally unstable system into a sustainable bio-based economy, okay? Today, what we are having is an unstable system. Unstable system means we are not focusing on a perfect utilization of the resources. We are only focusing on the prime aim is just to meet our demand, okay? We are not thinking about what will happen in the future if the way we are using our resources. To overcome this problem, industrial biotechnology is providing a platform, okay? If we look at biotechnology, biotechnology is the most significant and innovative sector, and there are large number of domains which are there in a biotechnology. And Tversky in 2000 developed a color code to differentiate main areas of a biotechnology, okay? So this is a color code which was given by Tversky in 2012 to differentiate the main areas of a biotechnology. If you see, there is a green biotechnology. Green biotechnology deals with your agricultural field, plant biotechnology, tissue culture, biopesticides, and biofertilizers. Blue biotechnology is a biotechnology which deals with your marine organisms, okay? Red biotechnology, hope you all will be knowing. Red means related to your biopharmaceuticals, like drugs, vaccine, antibiotics, medicines. So red biotechnology deals with that part of research. Yellow biotechnology deals with your nutritional biology, food and nutrition. Research are related to your yellow biotechnology. When we talk about brown biotechnology, brown biotechnologies are the biotechnology areas which relates with the challenges of the arid land, like phytoremediations, like desalination, and biofertilizers to improve the quality of the dry lands. So brown biotechnology deals with that. When we talk about gray biotechnology is related to our environmental applications, like it focuses on the solution to overcome the environmental pollutants. Next is your purple biotechnology. Purple biotechnology is a biotechnology which deals with your law, ethical issues, biosafety, biosecurity. And at the last, if we look at the white biotechnology, this is our industrial biotechnology. If we just look at all green, blue, red, yellow, brown, gray, all are, when we go up in the scaling up all the processes will be including in your white biotechnology. Like if you are creating a antibiotic at a lab scale, when you will be going for upscaling, that will be included into white biotechnology. If you're going for producing a biofertilizer at a lab scale, and then if you want to make at an industrial scale, go for a commercialization, that part will be covered into a white biotechnology, that is your industrial biotechnology. Okay. Similarly, all these colors of biotechnology, when you will be going for commercialization or upscaling your product, that will be covered under your white biotechnology or your industrial biotechnology. So let us talk about what is white biotechnology. Okay. In similar terms, as we have, I have mentioned in the starting also, development of a bio-based product using your microbes, yeast, plants, etc. So let us define it in a scientific terminology: application of efficient techniques at industrial scale to get desired end product. So your output is fixed, that is a desired end product. Okay. And for the human society, by using viable microorganisms, cells and tissue of plants or animals, or even certain functional components of their organisms. So when we talk about certain functional components, these are mainly your enzymes that are biocatalysts. Okay. And all these processes of getting a desired end product occurs in a fully automatic bioreactors. Okay. So bioreactors play a very important role in the case of industrial biotechnology. And in the bioreactor, you have to provide a controlled conditions. Okay, you all know that these microorganisms, cells, plants, animal cells have a specific growth pattern. Okay, and this specific growth pattern takes place at certain conditions. So you have to optimize those conditions, not at the lab scale, but at the bioreactor scale. Okay, when we work at the lab scale, the control condition will be totally different when we work in the bioreactor scales. Okay, so industrial biotechnology is the biggest branch of biotechnology. Okay, and if we go into the history of industrial biotechnology, you know the rudimentary industrial biotechnology dates back to 16,000 BC, when Neolithic cultures fermented grapes to wine. Then there were microbial yeast used to make a beer. Okay. In 1800, Louis Pasteur proved that fermentation was the result of a microbial activity. But in 1928, Alexander Fleming, you all know, extracted penicillin from the mold. Okay, so he extracted the penicillin from the mold. But the production of large-scale production of penicillin started in 1940. Okay, though it was extracted or it was researched in the 1928, but the large-scale fermentation or large-scale production of this penicillin, this wonder drug penicillin, started in 1940. Up to phase 1900, there was a there was a production of primary alcohol, vinegar, and beer. Fermented foods and milks were there. If we look at pre-90s, in the 1940, glycerol, citric acids were there. In 1940, after 1940 and 1964, single cell proteins were discovered from the algae and the yeast, and then amino acid flavors, then biopharmaceuticals, microbial polymers like xanthan, dextran, they were all reported during the period of 1980s. Post 1980s, there were a lot of progressive work has been done, and it's still going on. And there is a lot of changes have been incorporated, like there has been bioinformatics has been involved, synthetic biology has been involved. A lot of changes have been taking place, a lot of research has been taking place. And you know, today in the biopharmaceuticals, recombinant proteins, monoclonal antibodies, there are a number of bioenergy in the bioenergy sector. The industrial biotechnology is playing a very important role. So why industrial biotechnology is important? Okay. I was telling you that as we are facing the problem of depletion of resources, so we have to take a greener approach. Okay. Greener approach means you have to include three things. Your profit margin should also be there. Your planet should also be saved, and your people should also be getting benefited. Okay. And to meet this, you have to have a bio-based economy. So let me define you what is bio-based economy. It is a biological process. Okay. It is a simple biological process where you have your natural systems, your natural ecosystems, you use your natural inputs, and in the meantime, you minimize the amount of energy you are using, okay? And the waste you are generating in the process is acting as input for another process, okay? You're getting my point? So your economy is in the circular form, like nothing is the waste. You are generating something, you are getting your desired end product, and the waste which you are using, you are again using that waste as your input feedstock for another product development, okay? If by the use of industrial biotechnology, it has been found that 33 million tons of carbon dioxide production can be reduced. If we use industrial biotechnology currently, we can reduce the production of carbon dioxide by 33 million tons. Okay, so you can see that industrial biotechnology is an environmental friendly approach where there is a less waste, less energy, and also you can preserve your natural resources. Okay. Now, if we talk about people, the main important aspect is your economy, your process should also benefit the people. Means your economy should be there. Okay, economy should be maintained. So what industrial biotechnology do? It prepares create a society-based areas like job retention and job creation will be there based on the knowledge-based quality jobs. There will be reduction in the society on the fossil fuels. So people will be less using the fossil fuels, and they will be socially responsible for if they are using them, and thereby conserving them for future generation. Okay, so there will be employment generation because there will be new techniques to meet the future challenge. There will be a new jobs, and there will be an innovation. There will be a valuable resources for the future generation. And if we talk about the profit, see, any process is defined economical when your input cost is less in comparison. When you are putting a low cost, but you are getting you're getting a higher yield. Okay, so your profit should be economically viable and the novel product at a reduced cost. Okay, so for this you have to go for front of the pipe approach. There are two greener approach. One is the front of the pipe approach, and there is one end of the pipe approach. Okay, so let me define what is front of the pipe approach and what is end of the pipe approach. So in the front of the pipe approach includes development of products and processes which consume less energy and are eco-friendly and economical. Okay, so this is the first step. When you are going for scaling up of any desired end product, you have to put in the mind that it should consume less energy, it should be eco-friendly, and it should be economical. Okay, so front of the pipe approaches, initially when you're designing the process for upscaling your desired end product, you have to focus on the three parameters, that it should be less energy intensive, it should be eco-friendly, and it should be economical. Another approach is that end of the pipe approach. Sometimes what happens that you are producing something chemically and you are generating a lot of chemical waste. Okay, so what end of the pipe approach for the industrial biotechnology is that you include processes that deals with the treatment of waste already generated. Okay, so end of the pipe approach is that in the process, like in the case of textile industry, lot of dyes are used. Okay, so when these dyes are used, lot of dyes comes into the wastewater, and these dyes, if they are taken by human body system, they can lead to mutation and they can hamper our DNA. So what you have to do in that case, before discarding this waste, you have to go for certain technology, like you can use certain microbes which are able to digest or degrade or remediate those dyes into a non-invasive, or you can say into compounds which are biodegradable, or they can use any enzymes which can degrade those harmful chemicals. So this is called the end of the pipe approach. If I give you the front of the pipe approach, a simple example is that, like in the case of your detergents, you know that the industrial biotechnology has, in 1970 only, they have solved the pollution of water pollution problem, where the laundry was using a lot of phosphates. So by using the enzymes, by replacing this phosphate with enzyme, they found that there was a better stain removal from the clothes, and there were under waste which was generated earlier with a lot of phosphate was not the was decreased, and there was no polluting bio-based additive was there. So in this way, this was as an example of the front of the pipe approach. Okay, is it clear to you what is front of the pipe approach and what is end of the pipe approach? Okay, so let us come to this figure. So now you can understand. So now you can understand why I have given you this front of the pipe approach. See, if people is having a knowledge based, if they are having a knowledge regarding, if their people are having exposure to industrial biotechnology, they can design a process based on the front of the pipe approach, which will be economically viable and novel products can be produced at a reduced cost. Okay, now sometimes it can happen, there is a process which has already resulted in your waste, and now you have to use this end of the pipe approach to save your planet, to save your people. So what you have to do, you have to apply an end of pipe approach where you will be reducing the generation of the waste. So what are the key players of industrial biotechnology? You all know fermentation technology is very an old age process where we use microbes to generate our desired end products. Okay, so in the fermentation technology, there is a main basic step is your microbial growth. Okay, and then once your microbial growth has taken place, you go for the extraction of your compound desired end product. The extraction of desired end products and purification is included in your downstream processing. Okay, so extraction and purification of your end product comes under your downstream processing, while microbial growth, enhancing your microbial growth to get a better yield, comes into your upstream processing. Okay, and this fermentation technology requires your proper design and development of your bioreactors. Fermentation technology, use of a whole organism, we use whole organism, like your whole cell has been used. In enzyme technology, industrial biotechnology, in the enzyme technology, we use a biocatalyst. Okay, so these biocatalyst can be your source, can be anything. It can be a microbial source, it can be a plant source. So enzyme technology, we use enzyme. The enzyme can be a crude extract where the purification has not been done. It is a mixture of all type of compounds, or it can be a purified enzymes. Okay, so using an enzyme technology, you can go for upscaling. So can you give me a simple example of fermentation technology, which you are using products in the day-to-day life? A simple example of fermentation technology, the product you are using in your day-to-day life, right? Good. Fine. Okay. Right. So this is a fermentation technology, simple example of fermentation technology. Right. Yakult, Yakult, you will be hearing of Yakult, which is a marketable product, is also a type of fermentation technology where they are using a probiotics. Okay. Now, can you give me enzyme technology where enzymes have been used? Enzyme technology, I'm talking about enzymes, right? Detergents, okay, detergents are also there. Am I right? Am I lazy in biscuits? Okay, so there are different type of enzymes, leather making, right? Food flavor industry, right? Paper industry, soap industry. Okay, so these are the industry where enzyme technology is playing part. Now, with the advancement, the plant biotechnology also became a key player in the industrial biotechnology. Okay, so plant biotechnology is, plant tissue culture. They have used a plant tissue culture, plant cell culture to produce a certain type of your desired compounds. Like Taxol is there, which is an anti-tumoric agent. Okay, so large scale production is done by this plant biotechnology using a plant cell culture. Apart from that, you can go with GM, genetically modified plants, to get your desired end products. Now, animal biotechnology also is used in the case of industrial biotechnology, and this is erythropoietin and antibodies are produced by using the animal cell line, okay, cell line culture. And the most recent development in animal biotechnology is our COVID vaccine. Okay, so they have used the vectors which are having your virus protein inducing gene in the adenovirus, and they have used in the cell line, human embryonic kidney cell line, and they have produced a COVID vaccine from them. Okay, so this is a simple example of animal biotechnology where large scale production of the vaccine of COVID is done. Now, let us talk about metabolic engineering. Metabolic engineering is a stream where we have targeted our metabolite. Okay, we target our metabolite, and to enhance its production or yield, we go for engineering. Engineering can be that you can use certain vectors to include this gene into microbes, and because microbes have a very high multiplication rate, and due to this high multiplication rate, you will be getting a high yield of your desired metabolite. Now, the term is nowadays synthetic biology is coming into the picture. In synthetic biology, there is automated system where we design our gene. Okay, we doesn't cut or paste any, we doesn't cut or remove any gene, but we design our gene. We have designed our promoter, we design our gene segment, and then we use these genes for increase in the production. So what is the mode of technique which is used in the industrial biotechnology? Extraction of desired compound, that is from whole cell or tissue, as we have seen it. It can be from a plant cell culture. Whole cell you can use if plant is producing it inside in the intracellular, or it can be from animal cell culture, or it can be a microbial cell culture, or it can be a biotransformation. Biotransformation means you are transforming a certain substrate into a certain product. Okay, so the substrate you are providing, and then you are focusing on the product. Okay, so transforming the substrate into a product, this can be done either you are using a whole cell or tissue. Whole cell means you are putting a whole cell directly onto the your substrate, and the cell is converting, because cell is having a certain set of enzymes, and then you are using converting it, or you can use enzymes also. So these are few of the industrial compounds or industrial products which has been produced, like bioethanol, L-glutamic acid, MSG, citric acid, L-lysine, vitamin C. So you can see the world production ton per year. The maximum is the case of a bioethanol, which is produced from corn or sugarcane. Okay. Next is your antibiotics, which are produced in bulk, or they are some antibodies which are very highly specific. So they are production per ton year is given here, and the very less in the case of your vitamins. So we were talking about increasing the yield. Okay, so how can you increase a yield? You can increase a yield when your cell is multiplying very fast. Okay, cell is undergoing multiplication very fast, then only cell will be able to produce a maximum yield. Okay, product quality. So in that case, you have to focus on, first of all, on your organism which you are selecting. Okay, so the organism should be your prime area, because you will be using a fermentation technology, and you will be using the concept of metabolic engineering. Okay, so let us start with your first is what substrate you will be using. Okay, so as we are working with industrial biotechnology, we will be focusing on using a waste. Okay, so that our process will be a economically feasible, environmental friendly. Okay, and it will be ecological balance. It will be maintained. So the biomass, which normally is used in the case of industrial biotechnology for producing a microbial cell factory, is basically are the forest residues or agricultural waste. Okay, and these can be generated from your oil crops, or you can from just rice straw, wheat straw is there. Then there are certain non-grazing cellulosic components are in, non-cellulosic plants are there. So these can be used as your substrate. So there is also a point which need to be considered when you're choosing a substrate, that your substrate should not be a part of your food system. Okay, like in the case of bioethanol production, which is a very used in the case of in the case in US also, and in Brazil also, they are producing ethanol from a corn and from the sugarcane. So the concept of fuel versus food is going on, because this sugarcane and this corn are a part of our food system. So in the way, if you're using them for producing a biofuel, that is not a feasible process. So for that case, you have to choose your substrate which is in plenty and it is not competing with your food system. Okay, so that why we have to focus on the residues, we have to focus on the agricultural waste. Now, if you are using these residues or waste, you have to go for certain pre-treatment, because if you know these waste are having a rich in your case of cellulose and lignin and hemicelluloses, which need to be pre-treated to get a simpler form of sugar that can be utilized by our microbes. Now, feedstock selections is depending upon 4 parameters, as I have told you. Cost. It is that if you're choosing a very costly feedstock, it will not be feasible to run a industry at a high scale up. Okay, now next is that your feedstock should be in proximity to your production facility. If your feedstock is like you're working in Andhra Pradesh and your feedstock in Kashmir, the more of your transportation cost will be included in that. Okay, so to when you are selecting your food stock, you have to take into consideration that it should be close to your production facility, your industry. It should be close to your industry. Okay, as I have told you, there is a food versus fuel is going on, so you have to keep in the mind that your feedstock should not compete with your food system. Okay, now you have to design a pathway where you have to get a desired product. So you have to choose your agricultural waste which will meet in production of desired product. Okay, it can happen that you are choosing a substrate which will not result into an end product. So you have to choose a pathway where you will be getting a desired product depending upon your feedstock. Once your feedstock is ready, when you have selected your feedstock, you have done your pre-treatment. Next comes the role of your microbe selection. Okay, so microbe selection is that you choose an organism, Okay, it can happen that your organism is able to produce the desired end product, but the yield is less. Okay, so in that case, you have to go for your metabolic engineering. Okay, now in the metabolic engineering, each cell is converted into highly efficient mini reactor. Means that in one step, the desired yield will be produced. That cell will be acting as a microbial cell factory. Okay, so what you have to do first, you have to understand the whole genetic sequence of the organism in which you are going to going for changes. Okay, next you have to go for genetic engineering. You have to incorporate your desired gene which you want to be produced at high yield, whose output can be produced in high yield. That you have to go for recombinant DNA technology. Next, based on that, you will be going for small scale screening. Means it can happen that in one case, if when you have going for the producing your recombinant or genetic modified organism, there can be different levels of yield. So you have to go for a selections of the cell line, which is giving you best yield. Okay, once you have selected the best yield cell, you have to go for small scale fermentation that we used to do in our lab, that is a batch scale. Okay, so next step will be you will going for your batch scale fermentation. If you are getting a best result in a batch scale, then only that your genetically modified microbe will be selected as a final strain. Okay, it can happen that during your screening to the small scale fermentation, your microbe is not giving your desired result. It can happen that this can be due to a process changes also, like sometimes you are not able to optimize the process condition also. It can be a result of that also, but for that, in that case, you have to go for process optimization like growth conditions, and then you will be selecting your final strain. Once your final strain is selected, now you will be going for maximizing your productivity, your titer, and your yield. Yield is depending upon the what amount of feedstock you are giving and what amount of product is generated. Okay, so it depends upon the feedstock which you are giving and amount of yield which you are generating. Okay, if you are giving a lot of feedstock and you're generating a small amount of product, you're getting a low yield, then that is not a feasible process. When we talk about the productivity is that in what time period and what amount of condition your media, you are getting a maximum number of productivity. So productivity is related to your time period. Okay, next is your titer value. Titer value is related to your purification step. Okay, so more the concentrated product is generated, the more beneficial, the more benefit you will be gaining in your industry. Okay, so after you have maximize your productivity, your titer value, your yield, now you will be going for the downstream processing. Up to this, when we were talking about bioprocessing using a bioreactor, using of recombinant strain, all are included into upstream processing. Okay, now when you have got your desired product with a maximum yield, maximum productivity, now you will be going for your purification. Purification means concentrating your desired product. This will increase your titer value. So more you are concentrated, the more is your titer value. Okay, and the advantage of using this downstream processing is that it will reduce your cost, because if you're transporting your things in a you can say a less concentrated form, it will be causing you more transportation. But if you're transporting something in a concentrated form, in a small volume only, you can get a desired amount, and where you are transporting, they can go for a dilution also. So downstream processing helps in a high titer value, and you can get you can remove the byproducts during the downstream processing. So in the way from there you can get polymers, you can use this your desired products in developing chemicals, or you can use those enzymes in the biofuel generation, or you can go for you can get a polymers also. Okay, so this is a simple process where microbes are using as a cell factory, and they are called as microbial cell factories. Okay, where fermentation technology and metabolic engineering are playing an important role. Now, another key player when we were discussing, we were talking about enzyme technology. So industry biocatalyst, you know, is our enzymes. Okay, and what the role they play. They are either in the form of crude form, or they are in the purified form, or they can be in the form of cell lysates, or the whole cell form can also be used to convert the product into industrial valuable forms. But the problem resides in finding a perfect biocatalyst. Okay, so a suitable biocatalyst for any particular process fulfilling is still criteria and is still a challenge task. And for upscaling of the process, we have to go for a certain process. Okay, so when we look for a dream catalyst, we have fixed our reactant and a product. We know the process. We know the we have to choose the biocatalyst. Okay, so first step is your screening. Okay, so screening means which microbes or which cell is going to produce your enzyme. The first step is your screening. Okay, so once you have screened your microbe, like it is a bacterial biocatalyst or it is a fungal biocatalyst, so you have to screen them. Okay, so now once you have screened, is other step is that will the enzyme will individually be resulting into a desired product, or you have to go for the concoction of the enzymes, like mixing 2 or 3 enzymes, and then you are going for the desired product, or you are going for step-by-step use of an enzyme. A simple example of step-by-step use of enzyme is, you know, that for in the case of bioethanol generation from lignocellulosic, we use celluloses, hemicelluloses step by step. Okay, we don't put cellulase and hemicellulose at the time. Step by step they are used. Okay, now once you have gone for your catalyst selection, next step is your characterization. You know that enzymes are active at certain pH, temperature, and at certain conditions. So you have to choose those, characterize your biocatalyst. Okay, so you have to understand its kinetics based on your temperature, time. You have to understand the reaction condition at which your biocatalyst is having a best conversion rate. And next is your structural information. You can go for the structural information of your enzyme also to understand the active site of your enzymes. Okay, apart from that, sometimes biocatalyst engineering is also done. Biocatalyst engineering is also done, and after that they are gone for the applications. Applications, when your application, you have to check for the stability, that your enzyme is stable for a longer period till your process of is going on. Sometimes cofactor are required, so you have to look for the cofactor regeneration. And that if you are working in a multi-phase system, you have to check that your enzymes are not getting you can say inhibited by certain products. So you have to look after these parts when you are working with the application. Once your application is done, then you will be going for your product recovery. Okay, so this is the process of industrial biocatalysis, where you first go for isolation of novel biocatalysts for industrial process to decide which is the deciding factor. Next, you go for the metagenomics if you are interested in the biocatalyst engineering, or you can go for the classical way of finding, like you can do a classical like screening and then you biocatalyst characterization, then going for reaction condition in which it is working best, like changing the pH, temperature, salts concentration, or you can study these parts also. And then you can apply, or you can go for a biocatalyst. Once you have got information of the structural information, you can go for biocatalyst engineering, and then you can go for the application. So this is the way where industrial biocatalysts play an important role because they are eco-friendly, they are cost-effective, and their long-term sustainability is there. As you know, in the chemical process, there is a higher reaction rate. There is a lot of waste is generated in the chemical process, in the chemical catalyst. Okay. So, what is a roadmap for a successful scale-up when you are working with your industrial scale? So first we work with at a lab scale. Okay. So once we work with the lab scale, we go for a pilot scale. So what is a pilot scale? Pilot scale is a small scale preliminary study conducted to evaluate feasibility, time, cost, scaling up, and unpredicted results. Okay. And after the pilot scale, the next is your demo scale. Okay. So demo scale is done at larger scale than a pilot scale, and it is a continuous process like a full scale commercial production. And in this case, you have to go for certain processes. Okay. These are few points which we need to consider when we are working with our lab scale to so the first step is you adjust your formula for larger scale. Okay. So when we work in our lab scale, we have a small media, we have a small formulation. So we are easy to handle the formulation, the change in pH, we can handle it. But when we are working in the pilot scale or demo scale, we have to check for the changes which are occurring in the media at the large scale. So you have to adjust your formula on the basis of the changes which are happening in the larger scale. Okay. Next is the selection of the right equipment. Okay. You know, the main important scaling up is your bioreactor. So you have to choose the best bioreactor which will be working in your system. Okay. And the best equipment will determine your product quality. Okay. What will happen if you are working with a submerged fermentation, like you are going for a submerged fermentation, and you are choosing a process in which your proper mixing is not done. Okay. So in some submerged fermentation, you have to have a proper agitation. So if you are choosing an equipment which is not having a proper impeller, so your production yield will be hampered. Okay. So you have to choose your right equipment depending upon your desired end product. Next is the most important is determining the cleaning and sterilization need. Okay. So equipment should be easily able to be cleaned and sterilized. Okay. Next is identifying relevant parameter codes. The parameter codes which we work in the lab scale are not different when we work at the pilot scale or demo scale. So what we have to do, we have to identify now the next level of parameter codes, and we have to provide a process optimization we will be doing at this level, and to get a best condition in which we are getting a higher yield. Once you have optimized your process, your equipment should be such that you can include those changes in your instrumentation part. So these are the steps which will be included for successful scaling up. To give an example, to make you understand, let me give you an example of astaxanthin product, which was produced successfully from an yeast by Mexico toll manufacturing plant for more than 2 years. And the production yield was 7 gram dry weight per liter. Okay, so you can understand a company is producing, like Mexico toll manufacturing plant is producing 70 gram dry weight per liter for more than 2 years. Okay, so what happened? The UK company approached and they wanted to upscale this process. Okay, so Mexico was producing a 50,000 in bioreactor, using a 50,000 bioreactor, and UK company was going to use a 220,000 of bioreactor. So this astaxanthin cell growth was happening in 3 days, but intracellular astaxanthin accumulation was having after the remaining day, that is fermentation time was about 8 to 10 days. Before transferring these process to the UK company, they replicated this at a US lab. So at the US lab, when they were working in the lab scale, they got a better result in the case of Mexico. Okay, so but there was a product concentration variation was there, but the yield was more in the case of US lab. So the technology was transferred to the UK company, but what they found that yeast growth was normal. That yeast was growing at this normal rate, what was happening in the case of Mexico, but astaxanthin concentration was much lower. Means astaxanthin was less than 70 gram dry weight per liter. And in the meantime, they were also finding a contamination in their fermented batches by foreign bacteria and yeast. So in the process of these, their plant was losing a lot of money and were running at a 40% of the capacity. So you can imagine that in the process of scaling up from, there was a manufacturing plant which was already producing 70 gram dry weight per liter, but when it was converted to UK company, there was a same yeast growth, but astaxanthin concentration was lower. So what could be a reason why it went wrong in the case of UK company? The problem could associated was bad starter culture. Okay, so starter culture, you know, that should be in a better growth phase. Then only it will be able to adapt to your system and will be resulting your perfect yield. So what they did, the company shipped their starter culture to the US, and they validated that it is the starter culture the reason behind there is a loss in the product in the concentration. They also checked the fermenter mixing differences. Okay, so for that they recruited expert to study the mixing problem. Like it can be a reason that the proper mixing has not been done. That's why the product yield is less. Third, they checked about bad processed water in the UK. That can be the processed water which they are using for the production can be the reason. So they switched to the RO water. Okay. And next, to remove the contaminating microbes, the UK plant succeeded in reducing the extent of contamination with a small improvement in the production unit. So they used this solution, but then also there was not a perfect result they obtained, and there could be 100 possible root causes behind this concentration, less yield. And there we, as a process engineer or as an industrial biotechnology, we have to take a role to understand the mechanism of lab scale to pilot scale to demo scale. If you have a proper understanding of how you will be moving from pilot to demo scale, then only you will be able to understand the possible root causes behind your lack of yield. Okay. So this was about your basic understanding of industrial biotechnology. Now let me give you an Indian scenario of industrial biotechnology. In India, there are biotech startups are more than 270 companies are there. Biotech companies are greater than 250, and FDA approved plant, that is a US based FDA approved plants are 665, and manufacturing plant units are 1400. Okay, if we look at the percentage share of industrial biotechnology in India, there is biopharmaceutical companies which make up to 58%. Okay, in India in 2020, and this pharmaceutical segment is the largest sector of the Indian biotechnology sector, and normally in India consists of 250 to 300 companies, which account for 70% of the product of store vaccines, therapeutics, diagnostic, regenerative medicines, and medical technology. So you can understand that these biopharmaceuticals are playing a very important role. And the major companies, you know, Biocon, which is a biopharmaceutical company, which is providing you all types of products. Now, if we look at the bioagriculture is the third largest sector of the Indian biotech industry. And apart from providing biofertilizers, biopesticides, it is also contributing to the growth of Indian agri-growth market. And it contributes to the 15% of the segment of the industrial biotechnology. Next is a bioservices. Bioservices is becoming the next big domain in the biotechnology industry, which includes contract research, preclinical and clinical trials, and is set up to play a key role in shaping Indian rapidly developing bioeconomy. So bioservices, like if you can see our biopharmaceutical companies are getting attached to the different types of foreign companies for clinical and preclinical trials, like Serum Institute is getting attached. So these are types of the bioservices which our country provide to other companies from the outside foreign countries in the terms of preclinical and clinical trials. Next is your bioindustrial, which contributes to 9% of the total of industrial biotechnology segment. And this mainly comprises of enzymes that have multiple industrial uses and in the generation of bioenergy. So these also includes our food and nutrition sector and leather and green chemicals also. Bioinformatics is the smallest contributor in the Indian biotechnology industry. In recent, it has shown a great potential to grow, but it has only shown 2% role in the industry biotechnology segments in India. So let us start with our case studies related to industrial biotechnology. Hope till now you have got a proper knowledge about what industrial biotechnology is. It is clear to all of you what till now. Should we proceed to case studies? Okay. So let us start with our biopharmaceuticals. You know, biopharmaceuticals is a very big segment of industrial biotechnology. You know, COVID vaccines are already in the market, but most of the COVID vaccines are derived from human embryonic kidney cells or PER cells, which is also embryonic cell line. Now, the plant-derived COVID-19 vaccine is in the phase 3 clinical trial. Let me give you details of this plant-derived COVID-19 vaccine, which is at. So Medico biopharmaceutical company headquartered in Quebec City and GlaxoSmithKline, GSK, you all know it's a well-known company. So they have gone for a plant-based vaccine production technique. Okay. So Medico is providing your plant-derived COVID-19 vaccine, and GSK is providing you adjuvant AS03, which is an oil-based adjuvant, which is given to maintain the integrity of your vaccine. So what Medico is doing? Medico is producing a coronavirus-like particle technology, and he is using a recombinant spike glycoprotein, okay, expressed as a virus-like particle co-administered with the GSK adjuvant, pandemic adjuvant. So 2 doses of this CoVLP are under study, where they use 3.75 microgram of CoVLP are administered 21 days apart. So these trials have been in the phase 3. And what difference it creates, like in the case of this vaccine, generates a more about 10 times higher antibody response in patient recovering from the virus. Okay. And apart from that, these vaccines stimulate cellular immune response of T helper 2 cell, that is production of interferon gamma and interleukin 4. In comparison to COVID-19 vaccine, which is given to our patients nowadays, it stimulates T helper 1 response. So these vaccine is inducing the T helper 2 type, while our COVID-19, which is in the market, stimulate T helper 1 response. And the one advantage of using this type of vaccine, plant-derived vaccine, is that these hold an advantage that it can be rapidly scaled up. And because we are using a plant, we are not using any cell line, which requires a proper conditions for a growth. And they can be stored between 2 to 8 degrees Celsius. So you can see that in the COVID vaccine, the major important point is the storage of the vaccine. So in the plant-based production, advantage is that it can be stored between 2 to 8 degrees Celsius, and in a varied environment, it can be useful. So how they have gone for a production of this virus? So they have used a Nicotiana benthiana, which is a relative of tobacco, as a bioreactor to manufacture non-infectious coronavirus particle CoVLP. Okay. So the plant is not genetically modified. They have transfected the plant with a full-length SARS-CoV-2 spike glycoprotein. So they are not modifying the your plant, but they are transfecting it with a full-length SARS-CoV-2 glycoprotein. Okay, so what they are using a disarmed agrobacterium vector to deliver the foreign episomal DNA into the plant cell nucleus. Okay, so they are using a Nicotina benthica vethimiana with using a transient transfection technique with a disarmed agrobacterium mutations vector to deliver this foreign DNA into the plant cell nucleus. The advantage is that in the case of recombinant using this type of vaccine is that the recombinant virus-like particles self-assemble. So the major problem is that they undergo self-assembly and they board off from the plant cell surface. So they accumulate in between the plasma membrane and the cell wall. So the accumulation of virus-like particle is between the plasma membrane and the cell wall. Now, what you have to do, you have to take the aerial part, that is the leaves. Leaves are taken, they are blended to extract, and the purification of the VLP for the use of vaccine is done. Okay, so in this case, recombinant VLP is not infectious because we are using only the SARS-CoV-2 spike glycoprotein. So the core genetic material is not there, only the SARS-CoV-2 spike glycoprotein is there. So it is a non-infectious and it can stimulate your immune response. So this is the smallest, you can see this is a corona SARS-CoV-2, is a coronavirus, and this is a medical plant-derived virus-like protein of SARS-CoV-2. You can see this has a nanoparticle, has a size of 8 to 120 nanometer, and the SARS-CoV-2 have a lipid envelope derived from plant cell membrane. So these CoV-2 have a lipid envelope, and in this the S protein trimer appear to be anchored. So this is a size of your medical plant derived virus like protein, which is lacking your genetic material, which is having a lipid envelope derived from plant cell membrane, and the S protein trimers appear to anchor to this lipid membrane. Okay, so in this way, medical or plant derived virus like protein is in the clinical phase trial. So it will be getting approved. Now, apart from that, the Kentucky Bioprocessing is a company which is also working on COVID vaccine derived from plants. So these plants are also using an agrobacterium, and they are genetically modifying this with the coronavirus. They are using they are exposing 7 days to the agrobacterium, and after that they are extracting and purifying. So they are getting a 99.9% of their pure protein. But the problem in Kentucky Bioprocessing is that they are purifying the viral protein, and once they have extracted them, they are going to chemically attach them. Okay, so each of these components has been manufactured and purified separately. So all are the glycoprotein, all are the proteins of the SARS-CoV are expressed individually. Then they are chemically attached to form your individual virus particles. Okay, total protein. So in the case of your Medico, you can see they are getting a whole S protein trimer in the form of lipid membrane, but in them they are individually expressing the protein, taking out them, purifying them, then they are fusing them, then they are attaching them to each other by chemical modification. Now let us take another case study of pharmaceutical, that is your insulin production. You all know that insulin is a relatively low price drug. However, with the increase of diabetes in the case of in the global scale, the demand is more, and normally the recombinant human insulins are normally used, and two types of processes there. The first process is E. coli as an expression host, where the insulin precursors are formed as inclusion bodies, and which requiring solubilization and oxidation refolding, which is done manually. And then there is a yeast-based expression system, where the insulin precursor is directly secreted in the culture into correctly folded conformation. So the half of the world insulin supply is derived from Saccharomyces cerevisiae, where yeast-based expression system is done. In E. coli is that there is insulin precursors are formed in inclusion body, and once you have to purify, you have to go for oxidative refolding. So in these two type of expression host, you can see that yeast base is better because you can get a correctly folded conformation of insulin, and which can meet the demand of the at a larger scale. If we look at case study 3, this is the industrial scale production of artemisinin. You all know there are a lot of anti-malarial drugs in the market, but the P. falciparum, that is Plasmodium falciparum, is showing drug resistance against most of the drugs. So the most effective malarial treatment is now based on artemisinin-based combination therapy, and artemisinin is derived from sweet worm plant, Artemisia annua by TUU, and he got a Nobel Prize for this in 2015 for her discovery concerning the novel therapy against malaria. So he got a Nobel Prize for this artemisinin-based combination therapy for malaria, and artemisinin has been shown to be efficacious against all multi-drug resistant of P. falciparum. So in this case, how we will produce artemisinin? You know that there is a problem associated with natural processes, like leaves of tree were subjected to organic solvents like petroleum ether, and the extraction process is long, energy consumption is high, and production is low. So if you extract from your plant, you have to use those solvents like petroleum ether, which are not food grade solvents. Next is your extraction process is long, energy consumption is very high, and productivity is low. So the solution is semi-synthetic artemisinin. So for that, the production of artemisinic acid, that is the precursor, is done through fermentation. So, and once your artemisinic acid is produced, you go for the synthetic transformation of artemisinic acid into artemisinin via photochemistry. So here you are using a concept of semi-synthetic, where you are producing a precursor to fermentation. And once your precursor is formed, you are going for the chemical modification through photochemistry. Okay, so in this way, you are meeting the demand of artemisinin. Next is your, let us talk about enzyme technology, flavorzyme, which is a biotechnological route which is used in the flavor industries. So flavor enzyme is a commercial available proteolytic enzyme, which is prepared by Novo Nordisk Bioindustrial. So it gives a meat-like processing flavor when you use on a defatted soya meal. So you get a meat-like flavor when you use it on a defatted soya meal. And from cheese also, you can get an intense cheese flavor by using this flavoroenzyme. Okay, so you can understand that these flavors can be generated by the use of enzymes, and these flavorozyme is sold as an industrial peptidase, which is have a peptidase activity. It is consist of 8 peptidase enzymes, and they are derived from Aspergillus oryzae, which is a fungi, and is used in for various industrial and research applications. Okay. Apart from that, let us talk about detergent enzymes, which are used for the biotechnological routes of stain removal. The enzymes used are amylase, cellulases, protease, and lipase. And nearly 10% of the detergent mass is made up of your enzymes. And protease acts on a stain containing proteins like those of grass and blood. Amylase, you know, for works on the stain like sauces and ice cream. Lipase removes your oily stains. And cellulase minimizes color loss from the cloth and keep it soft. You know that there are a lot of products in the market like Ariel Matic front load, which is using enzyme technology. Surf Excelmatic liquid is also using enzyme technology. Henko stain care is also using enzyme technology to for your stain removals. But the certain problem which is associated with the use of enzymes is increase the cost of your washing clothes. For example, like Surf Excel Automatic and Henko is 2 to 5 times costlier in comparison to that of non-enzymatic ones like Nirma and Vim. So you can understand that for use of enzymes, can increase the cost also. Now let us talk about our fifth case study, like it's your biofuel, or you can say a bioenergy area, where I will be talking about the ethanol production. You know, biofuels can be of different types. It can be liquid like bioethanol, biodiesel, bioethanol, or biofuel can be gaseous, a biomethane, and all these types of biofuels can be produced at the industrial scale by the use of microbes and enzymes. Like in the case of biodiesel, algae are used, oleaginous microbes are used, oleaginous fungus are used. And in the bioethanol and the biobutanol, we are using lignocellulosic biomass for production of your biofuels. And we talk about the gaseous biomethane. You know that anaerobic digestion is a very important part of biomethane production, where microbes are playing a major role. So let us talk about the ethanol. That is the first generation ethanol, which is produced from either sugars or starch. Okay. So sugar-based ethanol plants, which is very much running in the case of Brazil, is from sugarcane, where you use a feedstock of sugarcane. And starch-based ethanol is used in the case of US and also China, Canada, France, Germany, and Sweden, where they are using a corn. Okay, so corn is used as a feedstock in the case of starch-based ethanol, first generation ethanol. Okay, but the problem is that in the process of generation of first generation ethanol, there is all the sugarcane and corn are part of our food system. So the fuel versus food is concept is getting emerged. So in that case, we have to go for other options. So first, let me tell you how this first generation ethanol takes place. First is your storage. You store your grains. It can be corn or sugarcane. You go for milling. Milling means you are reducing the, you are increasing the surface area, you're reducing the particle size. Okay. So you are in the milling, you are reducing the particle size. Then you are going for liquefaction. Liquefaction and cooking means you are generating the sugars or in the or starch. Okay. And next, after the sugar and starch are generation during the liquefaction process, you go for the fermentation using the yeast, and then you get ethanol. You go for distillation to get your distillation to recover your ethanol, and then you transfer it to the fuel market. The solid which is generated as a waste can be used to the feed market for the generation as a biofertilizer. So this is a first generation bioethanol. To overcome this problem of related to the fuel versus food, the next concept comes of the lignocellulosic biomass. Okay, lignocellulosic biomass are the biomass which is consisting of cellulose, hemicellulose, and the lignin. Lignin is a polyphenolic compound which is formed in the secondary cell wall of plants, and they are resistant in nature. They are not easily degradable. So for that, you have to go for the pre-treatment. So the examples of the lignocellulosic biomass are, some are edible by grazing animals like raw straw, maize straw, wheat straw, rice husk, jute waste, oil seed waste. And if we talk about non-edible by grazing animals, there are Lantana, Camera, Zetropa, water hyacinth, Bambusa bambus, Datura. These are all type of non-edible grazing animals which are also present as a lignocellulosic biomass. So this is your lignocellulosic biomass, consists of 23 to 30% of your hemicellulose, 38 to 50% of cellulose. So you can say if you digest your cellulose, cellulose is made up of a glucose unit. So if you can treat this cellulose with an enzyme, you will get a lot of glucose, and from there you can go for the fermentation and you will get ethanol. But the problem associated with lignocellulosic biomass is your lignin, which is resistant, which can't be easily degraded. To overcome this issue, there is a delignification process is done, which is a biological treatment using a lactase. Lactase is an enzyme which can degrade your lignin, and there are various types of lactases, that is yellow lactase and blue lactase, and they have a different structural conformation also. So either you go for this biological treatment using enzymes, or you can use these microbes also to digest your lignin molecules. And once your lignin has been digested, then you can use this substrate to go for a pre-treatment to digest your cellulose with help of cellulases and hemicellulose with the help of your hemicellulases. And once you get your digested cellulose, you can go for fermentation, and fermentation by using your yeast. Then you will be going for distillation to recover your ethanol. And once you have recovered your ethanol, you can directly go to use it in the combination. And the feedstock, which is the waste which is left, can be used as a biofertilizer. So this is all a bio-based economy process where nothing is generated as a waste. Everything has been optimized to be used in a proper condition. Now, this was all about industrial biotechnology concept and the potential which it's still holding. Now we will talk about the scope and opportunities. What is the scope and opportunities? There are various job prospects and the career opportunities. Like in the government organization, HPCL and Indian Oil are having their bioenergy sector area where you can work. There are government organizations like various DPT is also sponsoring your various projects where you can work. The private research organizations like Reliance Life Sciences and Hindustan Levers, there also you can have a job opportunities. You can work at universities, college, academic institutes like IIT, ISSR in their projects. Biotechnological companies, you can work in pharmaceutical and chemical companies, agriculture and food companies. So these are the areas, these are the companies where you can join your job. So what are the job roles which you will be offered? You can be joining as a technical officer, you can be joining as a scale-up engineer, you can be as a project manager, you can be a quality control executive, a quality analyst, or a process development scientist, or a formulation R&D scientist. But this all will require a certain experience or certifications for your before you join as in these roles. So what is a desired candidate profile? You should be a BTech or master in microbiology, biotechnology with certification, bioinformatics, industrial biotechnology, etc. You should be having experience in fermentation techniques, experience in fermentation kinetics, downstream processings, or you can handle a fermenter. Previously, you have had a fermenter of 15 liter, 100 liter, and 1000 liters. So the main is that you should be having a proper knowledge of fermentation technology, downstream processing, then only you can apply for these job roles. Apart from this company job roles, you can join a teaching positions based on your qualification in specializations, and you can be a project coordinators or scientist positions, but it will require that you should have a degree as MTech. You can be a PhD or a postdoc. For teaching position, PhD and postdoc is very important. For project coordinators, you can be MTech, and for scientist position also PhD and postdoc, the doctoral degree is very important. So this was all about your industrial biotechnology. Thank you, and kindly give your review on this topic. We would love to hear about you. Let us have some questions and queries, which is there. Hope till now everything is clear to you all. kindly give your review. I'm sharing the review link. Kindly give your review because your review matters. And so let us have some questions. There is a okay, so let us have some questions. I have given you the review link. Okay, so let us have some questions. What will be the efficacy of plant-derived COVID vaccine? See, efficacy depends upon the clinical trials. Clinical trials are going on, and you can say based on the clinical trial only, the clinical trials are done in phases. So in phase 1, phase 2, this plant-derived COVID vaccine has shown a better result in comparison to COVID-19 vaccine, which is in the market. That's why they have entered into the phase 3 trial. So you can, this efficacy can only be can be said after all the clinical trials have been done. Okay, so it depends upon the clinical trial results. If you have any related, you can just go and check on Nature. This article has been published in Nature, so you can have detailed knowledge about the clinical trials related to it on this article. You can just type this COVID-19 vaccine in tobacco plant, and you will be getting a whole idea in how this efficacy has been done in phase 1 and phase 2 trial. Now I'm doing my final year of BSc Biotechnology. So can I get a job in pharmaceutical industry after my BSc, and if possible, can you suggest such type of industry? See, Manu, the industrial biotechnology or pharmaceutical industry, experience matters, and at the level of BSc Biotechnology, you will not be having such amount of exposure. So I will say you go for certain certification courses, or you can go for a bit higher level of studies to join pharmaceutical industry, because I don't think pharmaceutical industry will be will have options at BSc level. Okay. Preetham has asked about why bioinformatics is why 2%, because bioinformatics is a nascent stage in India. There are less organizations which are working on the bioinformatic aspects of our microbes. So that's why in the case of India, bioinformatics is only contributing 2%. How biotechnology is related to law? You know that biosafety is related. Okay, like GM foods are there. So GM foods, biosecurity, biosafety, all these are related to your law, because some people are not accepting the GM foods, and also that when you work with designing any process, and it is individual, like if you have designed a process and it is a unique, you will be going for patenting that process, okay, or you're patenting that microbe which is you have designed. So in that case, patenting is related to your law, and this way biotechnology is also related with law part. See, in our country, the main important, as I have told you, the fuel versus food is going on. So you can't use a large section of food to produce a large amount of ethanol. So that's why the people or the companies nowadays like HPCL and industrial oil are using the waste to generate the bioethanol like lignocellulosic biomass. If you go and check on the industrial or Indian oil and HPCL also, they are having their bioethanol section, and they are using going for biodiesel production from using the waste. So that's why large scale production and uses of ethanol. The uses of ethanol is permitted in India. 10% blending or 5% blending is now permitted in India. So what are the qualification required for a startup? Qualification required for startup, Dinesh, you see, since you will be, I will say prefer you should have a basic knowledge of biological processes. It can be a BSc or MSc, and then after that you can go for your certification courses, and based on that, because for startup you should have a basic understanding of how the process is going on. So you should have a basic concept of microbiology, bioprocessing, and scaling up processes. So for that you should be having a BSc, MSc or MTech degree. Say Adarsh has asked about drug development is a type of molecular docking and comes under bio, why there is a less chances of bioinformatics in India. See, anything when it is related to biopharmaceutical or drug development, that can't come directly into a commercial market. Okay, it requires a proper studying process, preclinical, clinical trials. Okay, and when we talk about bioinformatics, there are very large. If you can just see in India, the bioinformatics courses has come into picture in the last 10 years. Okay, before 10 years, there were no such course of bioinformatics placed in India. Okay, so you can understand the people who are working in the area of bioinformatics are very less. Okay, and if you are talking about drug development requires a lot of time for commercialization. Okay, so that's why drug development is not an easy process, and bioinformatics is now growing in India, but it will take time. No, Vidya, there is no any placement exams going on in industrial biotechnology. Which field of biotechnology has more jobs score on feature? Which field? I will say the best biotechnological is your fermentation technology and your enzyme biotechnology. This is a basic industrial process where they are used. I will say that after MSc microbiology, you go for a PhD if you want to work in R&D section. Chemical engineering has a wider scope in the case of industrial biotechnology. Industry, yes, chemical engineers are most required as a biomanufacturing scientist or a process engineer scientist, because when you work with at a bioreactor level, the chemical engineers play an important role. Yes, Namrita, plants can be mass produced through tissue culture. Either they can be through cell culture can be done, or through callus or through callus formation also, and inducing the organogenesis, you can mass produce your plants. So hope with this, Arya, you have, if less is the downstream processing is there, then more will be, no. The more downstream processing steps you are including, the more purifying you are your product. Okay. Crude extract, you know, when you take a certain product directly from your fermented product, that is called crude extract. Okay. When you ferment, when you take a microbe, when you grow it on a certain feedstock, when you extract from that fermented feedstock, you get a crude extract. From the crude extract, you go for a downstream processing step by step, and depending upon your nature of your product, like if it is a protein, you have to go for downstream processing different. If it has a different, then if it's a secondary metabolite, you have to go for a different downstream processing. So with each downstream processing, your purification will be increasing. Your enzymes or your metabolite will be in more purified form. Someone was asking about an issue in, there is no form showing in this link. If you have any problem in registering in the course, you can kindly contact, the number has been given. Okay, from there you can have it, or you can directly contact Learn to Upgrade portal. They will help you out. Okay, so if you are having any problem related to your registration, you can just contact them. Okay. Yeah, Diya Pradeep, you are having MSc chemistry, you can get a job in a biopharmaceutical company. Yeah, yes, you can. But you should have a proper concept. See, if you are because you are a chemistry student, you should also have a knowledge about the biological aspects also. So then, on the basis of that, you can apply in a biopharmaceutical company. Okay, then, thank you all. Hope with the presentation you have been benefited, and we look forward to have you all in our next webinar. And okay, thank you. With this, I'm just sharing you the review link once again. Kindly give your review. We will be happy to have your experience with this webinar. Okay. Okay, thank you. With this, I'm ending the session.